So type 2 hypersensitivity is defined as reaction by humoral antibodies that attack cell surface antigen on specific cells and tissues, hence causing lysis of the target cells. Pathogenesis. So in type 2 hypersensitivity, the immune system produces IgG or IgM antibodies that bind directly to antigens located on the surface of the cells or tissues. And these antigens, they may either be intrinsic, which are naturally part of the body, or extrinsic, whereby foreign substances like drugs bound to the cell. Once antibodies bind to these cell-bound antigens, they trigger cell damage through the following mechanism. Number 1, complement activation. So antibody binding activates the classical complement pathway, leading to formation of the membrane attack complex, which lyses the target cell. So the complement system, also known as complement cascade, is part of the immune system that enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism, promote inflammation, and attack pathogens' cell membrane. Number 2, opsonization and phagocytosis. Antibodies and complement fragments like complement 3B act as opsonins, marking the cell for phagocytosis by macrophages or neutrophils. Number 3, antibody-dependent cellular cytotoxicity. So the natural killer cells bind to the Fc region of IgG antibodies on target cells and induce cell lysis without phagocytosis. The Fc region, which is the fragment crystallizable region, is the tail region of an antibody that interacts with the cell surface receptors called the Fc receptor. Examples of type 2 reaction include number 1, cytotoxic antibodies to blood cells. For example, autoimmune hemolytic anemia, in which the red cell injury is brought about by autoantibodies reacting with antigens present on red cell membrane. Secondly, transfusion reaction due to incompatible or mismatched blood transfusion. Number 3, hemolytic disease of the newborn, in which fetal red blood cells are destroyed by maternal antibodies crossing the placenta. Number 4, idiopathic thrombocytopenic purpura, an immunologic destruction of the platelets by autoantibodies reacting with surface components of normal platelets. Second example, cytotoxic antibodies to tissue components. So cellular injury may be brought about by autoantibodies reacting with some components of tissue cells in certain diseases, e.g. in Graves' disease or primary hypothyroidism. So thyroid autoantibodies are formed which react with TSH receptors to cause hyperfunction and proliferation. Secondly, in myasthenia gravis, the neuromuscular disease, antibody to acetylcholine receptors of skeletal muscle is formed, blocks the neuromuscular transmission at the motor end plate, resulting in muscle weakness. Number 3, in male sterility, anti-sperm antibody is formed which reacts with spermatozoa and cause impaired motility as well as cellular injury.